Hi, my name is Sarah Toe, and I am a human biology major at the University of California, San Diego. And today I will be interviewing Dr. Amir Zarrinpar, also from the University of California, San Diego.
I'm an MD PhD, so when I went back to do my clinical work, I noticed that a lot of my patients were obese, and I was very dissatisfied with the treatment options I had for them, which was essentially nothing that I thought was actually going to help them. And I actually went to a talk by Ron Evans, and during that talk, he just blew my mind with what you could do with genetics and molecular biology to affect metabolism and to understand it more. And at that point, I basically realized that we just have no idea what's going on with metabolism in general. And then I spoke to one of my mentors who, after I told him that I was interested in metabolism, and he suggested that I look into Sachin Panda, who became my postdoctoral advisor, to look into his lab because he was doing some interesting work looking at circadian rhythms and metabolism. And I thought circadian rhythms was a nice bridge also from neuroscience to physiology. And it fit that gap between metabolism and neuroscience really well for me. The hypothesis for time-restricted feeding arose from just observing mice that eat normal chow diet. Mice are nocturnal animals, so they tend to eat their food during the dark period. People notice that when you give a mouse a high-fat diet, or in this diet-induced obesity model, they tend to spread that caloric intake between day and night, almost to the point where they're getting the same amount of calories during the nighttime as they are in the daytime. What we hypothesized was that it wasn't just the high-fat diet that was causing obesity and all the metabolic problems associated with obesity, but it was also the disruption of these normal feeding patterns. So in order to test that, what we did was we created the time-restricted feeding model, which is where we consolidated the feeding time to the nocturnal period, which basically means we only allowed mice access to food during the nighttime, during their natural eating time. When we did that, instead of these mice becoming obese, they were actually quite healthy, and this was despite them eating the same amount of calories and being just as active as the mice that had access to food all the time, the diet-induced obesity model. So we found that maintaining those natural rhythms was protective against the detrimental effects of the high-fat diet. The mechanism for time-restricted feeding is very complex. We don't understand it completely. But what we do know is that time-restricted feeding plays an important role in maintaining robust circadian rhythm activity. Our liver not only gets circadian signals from our brain, but also gets input from our intestines in terms of from our feeding and fasting rhythms. When these two signals are synced, it basically behaves in an energy optimal manner. When you give an animal a high-fat diet and you disrupt the feeding rhythms, then these two rhythms become disjointed, they become dampened, and they could contribute to dysmetabolism. And with time-restricted feeding, what we think we're doing is that since we're restoring the feeding rhythms and aligning these feeding rhythms with the circadian rhythms of the peripheral clocks in the peripheral clock in the liver, that we're again optimizing metabolism to what it's supposed to be. I think that the question really is is, what do we think is the entraining agent from the gut, or this entraining signal from the gut? From our eyes, we know it's light. So from the gut, what is it? Is it a specific nutrient or group of nutrients? Is it a bacterial product? What we do know is that when the gut microbiome is not around, circadian clocks are out of whack. So it definitely plays a role. There's no question about that. But we just still don't know what that entraining agent is. I think the thing that's really blowing our mind is that the recent activity in our lab suggests that our microbiome is continuously adapting to us, and that, for example, my siblings and I may have inherited a microbiome from our parents, but since my sister lives in Hawaii and I live in California, and we may have different diets, even though we may have the same species in our guts, the genomes of those bacteria are probably very different from each other because they've adapted. Mine have adapted to be specifically in me, and hers have adapted to be specifically in her and the environments that we are exposed to individually. And I think that just adds another layer of complexity, which should be, again, fun to figure out. You know, when I was in undergrad, and even when I was a grad student, I thought that all the most important questions had been asked. And now I just think that there's so many questions to ask. There's so many things we just don't know. Really, I think that the best way to find inspiration is to see what assumptions other people are making. And to really start questioning those assumptions. So for example, with time-restricted feeding, people assumed that it was the diet that was causing this metabolism and obesity. We suggested that it may be something else. With the gut microbiome study, everyone assumed that the gut microbiome was stagnant and didn't really change. We started to think, well, what if that assumption's incorrect? What would we expect to see? I think that when you start looking at what are the assumptions that everyone is making, then you realize what are the questions that are worth answering that's going to affect everyone's research. We can take that, keep on taking these things further. Like, for example, I mentioned in my talk, why are we looking in the cecum? And then we realized we looked at the cecum because that's what everyone else does, and we never really had a good explanation for that. So we decided to look somewhere else that made more sense to us physiologically. I think that to people who come into my lab, I usually tell them to assume that everything that they know about obesity or microbiome is incorrect, including whatever discovery they make in my own lab, until they can prove it to themselves that they're right. And I think when you are curious and you want to answer these kind of questions, you're not going to run out of things to study.